Recording in progress.
Okay, hi. Happy Valentine's Day, class. Okay, let's get rolling because we got a lot of stuff to get through today. It's Tuesday, February 14th. Got everything recording. And we got an activity to do today. So get your clickers ready. Get this off of here. Yeah, so we're going to be using our clickers today. I have student hours are today 2:30. I have a meeting at 3 o'clock. So they're actually after class 2:20 today, let's say until 2:40-ish. And then I got to run. But we have our Q&A session on Thursday, right? So I'm here to answer questions. Thursday before spring break. Yeah. Sure a lot of you are going to be here. Use the time wisely. Okay, I'm here if you have questions. And I've been spot checking answers to deep questions. And maybe I shouldn't be, but sometimes I am quite surprised at I think you guys are not really getting involved and engaged in this course. Watch out, it moves fast. Knowledge lost in the beginning, you got to build that foundation. It gets hard. So the reason why I'm saying that is I think a lot of the stuff I'm talking about in class, exactly talking about, they're not in the answers, or they're actually opposite in the answers. And so there's not the understanding there that I kind of hope I get a lot of yeses and people understand, but I do realize that out of I've never seen 300 people in this classroom, even though there's 300 and some registered in the class. I'm sure it's all the people that are showing up are the ones that are nailing those deep questions. Good job, guys. For those of you that are unable to make it at least book timing or whatever to ask me questions, there was a lot of clarification needed. Anyways, okay. Not going to preach. I already did. That was too much. Okay, moving on. This week, learning outcomes. We're going to talk about norms of reaction. What does that mean? Well, I guess this section's about the influence of environment on phenotypes. So the learning objectives describe some non-genetic factors that can influence phenotypes. So we know that genes do. Genes dictate a lot, but actually there's also a lot of non-genetic factors. In other words, environment and so on. Discuss how phenotypes are due to a combination of heritable, genetic, and non-heritable environmental influences. Okay, explain how multiple genes and the environment interact to affect phenotypes. That's complicated. Describe how we can distinguish genetic sources of variation. Explain how phenotypic variation can result in a combination of environmental variation, genetic variation, gene times gene interactions, and gene times environment interactions. All right, sure, that's kind of along the same thing. Interpret experimental data. That's an important one. Be able to, we want to build your data analysis skills. So whenever you see magazines, newspapers, whatever, reporting stuff, you can critically evaluate that stuff for yourself, because oftentimes what they're saying, not true. You kind of wonder, where are they getting this stuff from? But so we want you to be able to analyze the data critically and interpret. Interpret experimental data and explain the relative influence of genes, environment, interactions, and so on. Describe how variation can be measured. All right. This past summer, grew a lot of stuff, including Rondo peas in my garden, very small garden that I had. And the Rondo pea variety is green pea. Does that mean that every pea from my pea population was green? Go. Okay, 5, 4, 3, 2, 1. I'm gonna stop. No, okay. Initially, 94 had the right answer, 94% of the class. When we went down to 88%. All right, so 88% did say no. Although Rondo peas are green, there's still going to be some variation. So you all have experience growing Rondo peas. Yeah. What's that? You can get a potato. Okay. Well, so they highly vary in phenotype. That's what you're telling me. Okay, so I think you guys, you're smart. It must be the pre-class materials that you all did excellent, or you just very smart and observant. So yeah, these are called green peas. And what you're telling me is, yeah, there's going to be variation, just like us. We're humans. Look at all the variation we got in this. A lot of variation, right? And if there were clones or identical twins of humans, you grow them in different environments, they look different. Have you seen twins with Arnold Schwarzenegger? Oh, do you guys even know who Arnold Schwarzenegger is? He's kind of old. He's my generation. A little older than me, but you guys know? Watch Twins. Danny DeVito, Arnold Schwarzenegger. This big guy and his twin brother. Anyway, they were grown in different environments. Okay. Environments can have an effect. Okay, so this is a box plot. Who's seen a box plot before? Wow. Okay. I'm getting a lot out of my audience. Way more of you know what a box plot is than Arnold Schwarzenegger. All right. Oh, okay. Good. You're very educated. That's fantastic. This is a box plot. A box plot is great. It's like a bar graph, but it's beyond a bar graph. It actually tells you more information than a bar graph does. What it's telling you, you can see you got, it doesn't matter what this axis is, a number of coffees drank during the final exam week. Oh, wow. And this is I guess the students that were polled 15 in the week, two a day. All right. Anyway, so out of all the people polled, what this is telling us here, rather than just a simple bar with some error bars on it, we actually have this box. And the box is telling us about the distribution of the results. Okay, so the middle 50% of the values, so that is from 25% to 75%, all of these values, they fit in this box. Okay, the 25% be down here, and above 75% would be up here. And this is the middle 50%. Very cool. Okay, so it's telling you where 50% of all the values are localized. Okay, and so what else do we have on a box plot? We have a median and a mean. Yeah, I admit, I commonly forget what the difference. I reviewed it before I came here today, but what's the difference between a median and a mean? A mean is like an average, right? You have a bunch of values, 14 coffees, 16 coffees, 25 coffees, blah, blah, blah. And then you let's see, if you have 20 values, you're going to divide everything by 20 and get the average. That's the mean. But what is the median? The median is the one I always kind of forget. Yeah. It's yeah. It'd be 6. Yeah, right. So if you had 11 individuals that you made this graph from, 11, they all got different values. The value of number 6, and you rank the values. You rank the values from highest to lowest. The one in the middle, okay, number 6, that would be your mean. And so the mean is actually, it's actually important, right? Because averages can be skewed. If I have one student that just like, the student had 56 copies. That big value is going to skew our whole data set, our average in the data set. But the median would not be skewed so much because again, it's the middle number of all the numbers, right? So anyways, so the median can tell us different things. What else here? And then we have, so our whiskers are, they're generally about 1.5 times as long as the width of this box, generally speaking. That's our whiskers. And so you get your 25%, you're normally going to find most of your 25% here, and 75% and above are going to be up here. And then you have these guys way out here. These are your outliers. This is the 26 coffees. Your 50% of everything was right in the middle in this box. And then you do have your whiskers, which are, 25% up and down. Then you have one or two, there are no coffees. That would be me. I can't drink coffee. I'd be running, you'd be frightened. I'd be running around the classroom right now and just like super fast. No. So the outliers are the ones that they're very, very different from that data set. Okay. And they're still part of the population. We still are, we need to be recognized, but they're very different. Okay, so now let's look at some examples here about the P's. So what this is two varieties of P's. And this is a box plot again. We're looking at box plot. Okay, so we have the Rondo variety, which is a green pea, and the Frisson variety, which is a yellow pea. Okay, and so what's being measured here is it's chlorophyll B. This is the amount of micrograms per gram seed coat. So they took the seed coats off and measured the amount of chlorophyll B. Okay. What can you see about the green one versus the yellow one? Yeah. Very good, very good. So Christian gave us two things. One of them he's saying is, hey, look, the yellow one, it actually, they're generally on average has lower chlorophyll content than the green ones. Okay, maybe chlorophyll B is involved in chlorophyll B is a green pigment, might have something to do with the color difference, right? Yeah. And also not as much spread. You can see those yellow ones are they're pretty tight, and meaning that their box is quite narrow. The values fit generally within this small range here. Whereas when we go to the green ones, the rondos, there's a much larger range that we're seeing. Okay, so I keep doing that. Yeah, so that's the box plots, but
Here in no life.
yeah, okay, we'll get to it. We'll get to it. Okay, so go. How should you classify P with 60 micrograms per gram chlorophyll B? Five, 4, 3, 2, 1. Okay, so most people in class, interesting, it varies from year to year. Most people in this class, 68% of those 150 students that answered are saying that you would classify the P that has 60 micrograms a gram right here, that would be yellow. All right, okay. So you randomly pick 3 Ps from each Frisson and Ronda and P populations. How likely is it that they would be easily distinguished from each other in color? Five, 4, 3. Oh, I'm going to hold that a little bit longer because you guys are going in the right direction. No, wrong direction. 8, 5, 4, 3, 2, 1. Okay, good. Now, let's pause here. I'm not going to do that thing where I just keep going and going without giving you guys any answers. So you randomly pick 3 Ps. Okay, how likely is it that you would easily distinguish from how likely is it that they would be easily distinguished from each other in color? So your answers were you can easily distinguish Frisson and Rondo Ps. Okay, is that true? Can you easily distinguish the two? Just randomly pick 3 of them. You always going to be able to distinguish them? Okay. I'm seeing about 95% inertness where people are just like, hey guys, yo. Can you easily distinguish Frisson from Ronda? Always. Can you always distinguish them? Oh, no. Okay. Why not? How about somebody from the upper tier? Why not? Yes, thank you. Okay, there can be variation in each population. Look at this. Look at the, well, we're measuring chlorophyll A, I guess, on this. Chlorophyll A is also green, by the way. Measuring chlorophyll A, and we got these data. And then we measure chlorophyll B. We got these over here. It's also green. And so we randomly pick one out. If we randomly pick this one right here, that's an outlier. I'm not going to go there. What if we randomly pick one of these ones right here? Would we know whether it's Frisson or Ronda? There's overlap. There's overlap, right? You might not be able to easily distinguish the two all the time. Might be more prevalent that it's green if it's Ronda, more likely, but you can also have some Ronda that might appear yellow if they have the same amount of chlorophyll B as Frisson. That's what these ones do. This is a figure showing peas from several British seed merchants in 1901. That's where they got those. But anyways, very cool. And so peas that Mendel used weren't as easy to distinguish as the figures in textbooks would have us think. Yeah, so look at these peas. As found, these peas that were around the same time as Mendel. And can you easily distinguish which ones are green and which ones are yellow? If we did that binning exercise and you have two bins, green, yellow, do you think you get the same thing as your neighbor would get by sorting all these peas into green or yellow? No, it'd be pretty hard, huh? Some of them are just like, they're kind of partway in between. It's not easy to tell.
You see what I sent you?
And so, you know what, this is just like a kind of a sidetrack thing, but Mendel, whenever Mendel did his experiments a long time ago, there were people, whenever the results were discovered, I should say, whenever the results were discovered, there were people that were, wow, they're really, Mendel's results are very clearly telling us something. Wow, look at this. And so they were looking at the ratios and everything that Mendel got. I'm not going to downplay it. It was an amazing discovery. Led us to understand how genetics fundamentally is inherited today. This whole units, they didn't know there were chromosomes and they didn't know they were alleles back then. But the data apparently was a little bit too clear cut. And the people were saying at the time, like, hey, look, there is no, like, these peas, it's hard to distinguish clearly whether they are yellow or whether they are green. And so there were other scientists at the time that were not really keen on Mendel's results. They thought that Mendel may have not reported things clearly. So, and they thought that they were able to, they had the opinion that differed from Mendel that, there's a range and actually there's more to it than just genetics. They were on the track. Phenotypes are being impacted by the environment. Even though you have the same genes, they had a variation in the color. That all was kind of like, it didn't have a loud voice in the scientific community. I won't say suppressed, maybe even. Suppressed, and Mendel's findings were clearly amplified and everybody was talking about Mendel's results. So anyway, so we now know that Mendel wasn't, it's not as clear cut as Mendel had shown. It's not as clear cut as your textbooks show you, those beautiful cartoons of the yellow being very different from the green peas. I teach a plants class. Those pictures are in that book. It's not so clear cut, but respectfully so, they did come out with the general inheritance patterns, laws of independent assortment and so on from those. But it is more complex in reality, as we can see here. Okay, so we're going to look at a study, and it's researchers were studying how crops with different genotypes will grow as climate changes. You know, with industrialization, one of the major consequences of industrialization is the accumulation of CO2 in the atmosphere. And as it's increasing, right? So the experiment here wanted to show how that affects plants, how it affects plants of particular genotypes, specific genotypes. How does it affect their height? So one experiment involves growing crops in fields where special equipment adds CO2 continuously to the air surrounding the plants. And there's sensors there, and they're going to keep the CO2 at a certain level. You guys are probably like, well, no, they can't do that because it's all going to end up in the atmosphere and it's going to destroy the earth. And like, well, but they have to do these experiments to figure out what is going to be the consequences if we keep on track with this CO2 pollution. Okay, so they actually did this. They pumped CO2 into the fields and tried to maintain certain levels of CO2. And it was soybeans that they were looking at. Yep. Soybeans are economically extraordinarily important. You likely have eaten something today that contains a soybean component, or you've ridden in a vehicle today that has a component built from soybean. Soybean has literally thousands and thousands, tens of thousands, maybe even hundreds of thousands industrial uses, from building car plastics to being present in processed foods. Super important. So we want to figure out, that's what this study was showing. I wanted to figure out what is the effect of CO2 on soybean growth. And they're measuring height as a reflection of growth. Okay, so in 2004, an experiment, the height and maturity of different varieties of soybeans was measured after growth in fields. CO2 around these control plants was, the control plants had no CO2 added, and then other plants had CO2 added. So you had to control this one, which is the regular amount of CO2 that we have today, and then they have the plants where there was CO2 added. And so here, these are different genotypes. Clark that's one we work with sometimes. So anyways, Loda, Clark, Panna, different genotypes. Okay, and here is without CO2 supplementation, with CO2 supplementation, and that's what they predict the levels of CO2 would be in 2050. Okay, so what's gonna happen to the plants? Looking at these trends. So in groups of 2 or 3, prior to 2004 experiment shown here, similar experiments were done in a lab. Why was it important to carry these experiments out in a field? Okay, talk about it with your partners. I'll give you 2 minutes to discuss. Why in the fields? Okay. So what's the verdict? Why? Why? Okay, please stop. Why is it, why in the field? Why is it so important in the field? Why do you think they did it in the lab first and then they did it in the field? What's the point of pumping all this dangerous CO2 into the fields? Why? Yes. Outdoors? Okay. It has to do with the variables, yeah. Yeah, anyone, yeah. Yeah, they're both of these, they're very good explanations. Yeah, so it has to do with the variation, right? Okay. So, right. In a lab, we can, like in a growth chamber, we use growth chambers, temperature is controlled, humidity is controlled, intensity is controlled, CO2 levels are controlled. We can control all the factors and we can change one variable, like CO2 levels, keeping everything else the same. Essentially, what we're doing is we're isolating that variable so we specifically know the effect of the one variable. Now, that being said, so, because if I modify, okay, so I just changing one variable. What is the difference when I put this out in nature? Well, put it out into a field, this massive field, CO2 machines pumping out the air and stuff like. What is the difference? Yes. Very good, very good. In the field, there are going to be a lot of variables that we don't have control. In fact, the only thing that we are controlling is the CO2. Okay, so we work it out in the lab first, changing only CO2 and finding our CO2, when we change it in the lab, we find that this happens, maybe higher CO2, more growth. Okay. But it might not reflect what's happening in the real world, in nature. We may have all this great lab data. Yes, let's go do this. We bring it out into the field and nothing we've seen in the lab happens because you can have environment interactions, right? Variables could be changing in the environment. How easy is it to control the variables out in nature? If you're a farmer and you're growing the 2,000 acre field of these soybeans, can you control whether every single plant has the same water, light, temperature, so on? In this class, you are expected, you should be able to explain why experiments are done in the lab first, and then they're done in the field afterwards. Okay? Each one has pros and cons, and that was a beautiful answer I heard about the cons of doing. So we just talked about the cons of doing everything in the lab. We're controlling one variable. We may think we have solved everything. Okay, this is great. You know, CO2 does this. We put it out into the environment, and all of a sudden we find it doesn't behave like we were expecting because there could be other variables we didn't even think about that are present in nature that are even more important. So what? Okay, let's just get on with it. Let's go to the. What is the general effect of higher CO2 levels on the height of soybean plants? 5, 4, 3, 2. Oh, yeah, it froze up on me. I think everyone got on their devices and just, hold on, guys. I've heard of this happening before. Have you guys had it in class, mid-class, where a clicker just dies? I've never had it. I've heard about it. I've just never seen it. Okay, it's back up. 182 responses. Great. Okay, so most people are saying it's C. Okay, so what do we see? All varieties are taller, but so 66% of the class say C. All varieties are taller, but the effect is different on each variety. Okay. okay? Because when we increase CO2, this one only increased a little bit. This one over here increased a lot. Very good. 17% said A, 17% said B. Okay, let's try this again. The data above are from peas grown in controlled lab conditions. What would you expect the data to look like if they were grown in nature? Five, four, three, two, one. Again. It's frozen up for me. I don't know if it's frozen for you or not. Okay, so what's the answer? I can't see your answers. What do you think the answer is? C. A lot of people are saying C. So, okay, I can stop it now. Great. Similar to the results from the lab experiment, but with more variation. Very good. Does everyone get that? Okay, good. Not seeing anybody saying no. Okay, so environmental factors, why would it have more variation? Why would we see more spread? The data, those boxes would look wider, stretched more. The data in general will look more spread out. Why would that be? Yes. There's more environmental variables, right? So with maybe water varied and some of them didn't build as much chlorophyll because, you know, to do photosynthesis, to build all this stuff in the first place, you need to have water. The water oxidizing complex and so on, the oxygen evolving complex. Not to get technical and stuff we don't really know about. So there's going to be a lot of variables. And so the data, it's not as tightly controlled. The results will not be as tightly controlled. So we don't know in the field what it would look like, but we would anticipate there would be more variation because there is more variables. Very good. Okay. You conduct a study to determine the height of an influence of more, if height is influenced more by genes or by environment. Your results show the figure on the right. What can you conclude about the data? Is it more by genes or more by environment? Wow. Yeah, clicker's having some major problems today. Okay, let's just move ahead here. We can come back. So what would it look like? What would it look like if height was the only thing affected? What would it look like if height was affected only by genotype? Height is affected. So we got height on our axis here. And we got those two varieties. We got the XY and the XX. What would it look like if, I was asking you, what is this telling us here? But what would it look like if it was only affected by genotype? Can somebody explain that one to me? If I can, I'll draw, you just explain it.
It's amazing, bro.
Yes.
I'm a #1 advocate for Hogwarts. So you say
Okay,
now?
so you're saying that, so if it was only genotype, I'm gonna draw here, XX,
Chill.
and, just to be consistent with the colors.
It's $100.
Here for purple. And, this one here, let's say this one here is X.
But do I really want to draw?
So, see the nutrient, high nutrient, low nutrient? They're essentially having no effect. The height is going to be maintained. They're not affected by the environment.
You didn't want to drop $15.
Now let's look at B.
Better skin.
What would it look like if the height was affected only by environment? Only by environment.
the
So in
skin's
other
only
words, there's
15.
no genotype effect. There's only an environmental effect. Yes. Okay, they're going to be one on top of the other. And yeah, they're just low nutrients. So let's say that the two lines start low like this and they go high like that over here. This is going to be our XY. If I could draw.
Because I actually didn't want it.
Okay, and the red one.
That's different.
Okay, would essentially be right over top like this. Okay.
And Ali's the one who offered it.
Okay, so it's infected only by environment.
So, Ali,
So meaning the genotypes, there was no difference between the genotypes. That's why they're overlapping.
70-30.
And the effect was only for the environment, so they increased with the environment. Yeah, sure, good.
Yeah, what is that, $30?
Okay. Okay, so
Yeah.
the genotype difference, if there was a genotype difference, let's say, what does this
60-40.
graph tell us? Okay, is it genotype or is it environment?
Okay, yeah.
Oh, both.
Yeah, because I
Both.
actually want it.
Right, because the genotypes differ, and you can see that, look at this, the XY genotype, it actually grows higher, XY, I guess we're talking about humans here.
First,
The XY, so
me
these
finish
males
the
are
game by
actually,
doing
they're
something
generally
like this.
taller than the female, okay? So there's a genotype effect, and the environment, is there an environmental effect? Low nutrient, high nutrient. If there was no nutrient effect, this would just be a flat line. It wouldn't change with a different environment, but it actually increases. Okay. Is the environmental effect more for one genotype than the other? No, it's the same. Yeah, it's the same. Why do we say it's the same? The slope of the lines are the same. So we started out with a genotype difference here, and both lines have the same slope, and ultimately so the end of the environment, which is indicated by the slope of the line, because we're looking across two environments, actually had the same slope. So both genotypes are affected the same way. All right. Okay. You want a trip to a beach destination over reading week? Can I come? You spent a lot of time outside and end up with a nasty sunburn. What best explains this phenomenon? Which of the following is most likely? Okay, you get a sunburn. Five, 4, 3, 2, 1. Wow, crashing. Yes. Okay, what do we got here? Oh, wow. Oh, no, there's a lot of stuff going on here. So 46% of people are saying B, only environment affects your phenotype. 27% are saying this is an example of genotype plus environment. And other people are saying, well, another 25% are saying this is genotype times environment. Plus environment is like it's more than the sum of the two. Okay, we don't have any numbers here, but okay, so only genotype, no, almost nobody said only genotype affects your phenotype. Okay, so in this example, you went outside, one person, you get a sunburn, okay? Why is B the answer? Yes. Oh. Hey, you are correct in your, and I see this sometimes in the questions that, or the deep questions. You're correct in your logic, but given the information in the question, this was a little, I'm not saying none of the deep questions are trick. They're definitely not trick questions. But you have to take the information that's given in the question. And here we have no genotype effect. Why do we have no genotype effect in this question? Because we're not looking at two different genotypes. We're only looking at one person. So, sorry, no, but your rationale in reality, but we would have to look at more than one person to know whether there's a genotype effect, right? You look at one person, you see what happens to one person. So here, there was only an environmental effect. Okay. MTHFR is an enzyme involved in folate metabolism. In humans, severe deficiency of MTHFR results in very high homocysteine levels, impaired connective tissue, muscles and cardiovascular system, and infertility. Okay, so a lot of bad things happen whenever you have a deficiency in MTHFR enzyme. A mouse model of severe MTHFR deficiency was produced by knocking out the gene, okay? Heterozygotes were mated. They're mated to one another. Two heterozygotes mated to one another. They're mated to, and then they're going to produce, they're going to count how many mice are MTHFR negative. So how many have the knockout? And so we can genotype the pups at 3 weeks of age. That's whenever they do the counting, they find how many are homozygous recessive or how many have the homozygous knockout. What is the expected ratio of the pups from across of the two heterozygotes? Okay, go. Okay, 5, 4, 3, 2, 1. Okay, most of you are saying 25%. Clicker has frozen up. The other 36% are saying, most of 32% are saying 50%. How do you determine this from the beginning? Can someone tell me how? Okay, yeah. Punnett square. Punnett square, Punnett square. So let's look at the Punnett square. Let's do the Punnett square. I'm always, every year, I'm a little bit surprised at how we don't remember how to do Punnett squares, but that's okay. We will practice them here. So I'm gonna make a Punnett square. Punnett square looks something like this. And I'm just gonna write negatives and positives rather than the MTHFR, okay? So we're talking about this. So remember what happens in the Punnett square. What's the first thing that happens in a Punnett square? How are you gonna fill in the first thing? What do you put up here? I was pointing with my finger. Up here and here. What? Yes. The gametes, the genotypes of the gametes, the genotype of the egg and the genotype of a sperm, which is haploid. Okay, so I'm going to put haploid. So it's not possible for me to have a gamete plus negative. I'm just going to put the separate gametes. Okay, I got plus here, negative here. Plus here, negative here. Why is it plus negative plus negative? Because these were selfed. I said that these heterozygotes were mated. They're mated to one another. So each of them can either make a plus gamete or a minus gamete. Okay, follow? So now I'm just going to fill in the Punnett square. What? It's going to look something like this. Plus, where am I? I'm getting this plus from here, negative from down here, negative, negative. I was asking about this genotype. They produce this, okay? How many pups are going to be this? I don't know why 50%. 25%. The answer's over here, right? This guy right here. 25%. That would be, get comfortable with the Punnett square. If you are not, you will have difficulties later. We're going to talk about sex-linked things and all kinds of stuff. All right? Okay, so 25% is what you would expect. However, what the researchers found is actually 0. There were 0 pups of that minus genotype. What that means is that it actually found that it's causing male sterility. And so, but anyways, those pups, those minus pups, are actually dying. They don't reach the 3 weeks of age. And so the deficiency causes infertility. If they found out if they supplement a pregnant female's diet with betaine, it's kind of like the product of the enzyme. Okay, this is like being medical doctors here. They supplied betaine in the diet in the mother. So when the mother is ovulating and ultimately gets pregnant and it has like the embryo inside and everything, all during that process, they're supplying betaine in the diet. And that was able to cause some of the pups to survive. So it's an environmental factor. It's nutrition in the diet. Here's your survival rate. This is no betaine, and you see the pups. They're dying. They all die. They die. Pretty much all die. 20% survive. And then if we supply betaine in the diet, huge increase in the survival rate. Yeah. So MTHFR deficiency is linked to male infertility. To see if betaine supplementation of pregnant females affected the fertility of the male offspring, the scientists gave betaine to the males. Okay? In utero, during nursing, via the mother, and so on and so forth. So the betaine in utero, during nursing, via the mom, and for the rest of the life, long-term treatment, they gave them betaine in the diet. Which of the following graphs supports the hypothesis that long-term betaine supplementation improves male fertility only for the negative genotype mice? And we're showing you a sperm count here. That's just a reflection of fertility. So which one of these shows that it's actually, the betaine is actually improving fertility only of the negative mice? Okay, I'm going to start the clicker for you here. Give you a few minutes to answer. 5, 4, 3, 2, 1. Okay. The vast majority got the correct response. They said D. I hope it's not because I forgot to delete this arrow. That's hope that. Let's go through this because this is like the whole section you saw on the pre-class. It's pretty much all about this stuff, right? So let's go through these. What are we looking for? If it's only the negative mice that are going to be effective, the blue guys are the negative, and the red ones are positive. So what are we looking for if we say only the negative are going to be effective? Yeah. Excellent. We're looking for a graph where the positive is not affected. It's going to be a flat line. Remember, short-term treatment, long-term treatment, no change. And if you look here, this one doesn't have that. That doesn't have it. This doesn't have it. So you could quickly rule out the ones that don't have it, right? But you can see here for the negative, there's a change with the long-term betaine supplementation. You can see the bar actually increases. So we got more sperm count in that male whenever the long-term treatment is present. Very good, very good. Okay, so. Okay, which of the following graphs would support the finding that long-term betaine supplementation improves male fertility regardless of genotype? Gonna give you a few seconds. 5, 4, 3, 2, 1. Okay. I'm not able to see your results. It's crashing again. Oh, there we go. Vast majority are saying B. Excellent class. Good job. 67% saying B. Why are you saying B? Okay, so remember what we're looking for here. What's the finding supports that long-term betaine supplementation improves male fertility, so sperm count, regardless of genotype? What were we expecting to see? Yes. Very good. The fertility, the sperm count of both positive and negative genotypes are increasing. Okay, so the betaine supplementation was working for them both. What are we finding here? Yes. Fertility is going down for positive with long-term betaine supplementation in here. It's not changing for the negative. Okay, so you can go through each one of these graphs. And what about this one? This one's really weird. So it's going down for negative. It's going up for positive. Wow, it's beneficial to one genotype, okay, and not for the other. And how many times, like, okay, so this can be the case for drugs. Drugs are kind of like this, medicines. And now people are, the kind of state of the art in medicine is actually to look at people's genotypes and figure out treatments. Personalized medicine. You may have heard that term, personalized medicine. Okay, because sometimes for some people, a drug, depending on your genotype, it might help you, and sometimes it might actually hurt you. So kind of like in this situation. All right, got to move on here. Okay, so now we're really going to look at a different study. And this one's about, so researchers were interested in why some children develop conduct disorder, while other children do not. These researchers focused on the MAOA gene, which codes for an enzyme that metabolizes brain neurotransmitters. They studied 1,037 children that were followed closely from childhood to adulthood. The figure over here to the right presents their results on the association between conduct disorder, maltreatment in childhood, and the levels of MAOA gene activity in male children. Okay, so this is a percent of children exhibiting conduct disorder. Okay, and here we have low MAOA activity and high MAOA activity. According to the figure, which of the following is true for conduct disorder in male children 10 to 18 years old? Social conduct disorder is what? Five, 4, 3, 2, 1. I am really impressed Very good, class. Yeah. 58% in the end there. 58% chose F. This graph is showing us that this social conduct disorder is impacted by both environment and genotype, but environment has a bigger effect. Why do we say environment and genotype? You look at these bars here, same. So no maltreatment, they look the same, doesn't matter the MAOA activity. Probable maltreatment looks the same, doesn't matter about the activity. And then, yes. No worries, you only get points for participating. But that totally trashes my confidence because I was so happy everybody got that. Yeah. I'm assuming everyone who put E was actually going for F. It was the last one on the list. F is the right answer. F is right. Why is that? Why is that? Why are we, look, it's impacted by both environment and genotype, but more so via environment. Why do we say that? Yeah. Okay, so look at this. These are the different genotypes, right? And we only get a big change from the maltreatment environment when we have a particular genotype. Okay, it is dependent on the genotype. So yeah, so that's why the answer is F. So bigger, you can see generally there's not a lot of difference, but whenever we have that certain environment, it causes a major difference. Okay, it's an environment times a genotype. All right, so let's go on to this one. Which of the following figures best represented the relationship between genotype and environmental influences on the phenotype of social conduct disorder? So which one of these bar graphs represents this? Oh. Okay, 5, 4, 3, 2, 1. Bing. Okay, so most of the class said D. Does everyone agree, D? Well, it's actually 69% of the class, but that's more than they chose the other answers. Nobody chose E, which is good. Okay, so 69% say D. So why do we say D? What is it about this that tells us this is D? Yes. Guys, please. I get distracted very, very easily. Okay, please quiet down, please. Yes. Yeah. So if you look at this, right, so we have the high MAOA, that's this one here, and it's increasing, it's increases slightly between this no maltreatment, severe maltreatment, slight increase, and then this one here, the low MAOA, huge increase, right? And so what do you think about this? Why is there a difference in the beginning here?
820.
Tell me about that.
What about here? 5, 4, 3.
Someone from the back.
Kermit?
Why do you think that there is a difference right here on D? Is this accurately reflecting the graph or not accurately reflecting
If it
the
was
graph?
10, he wouldn't toss you back. Move, make space. Ew, there's jewel marks! That's so gross. That's so gross. That's actually gross, bro.
You know why?
No.
Because every time I put my head there, the thing shines right into my eye, and I can't go further. 'Cause-
Gotcha.
Yeah. So then I have to move
Ew,
here,
That is gross.
and then sometimes even on here,
Yeah.
like my head on here.
Okay, the difference
the-
here,
No.
we got a difference in starting points.
The-
What do you see over
the light is still
here
shining, so I have to put my head down
See,
here.
what don't want to go
that you
there
don't
anymore.
see over
Stop.
here?
It's fine.
Somebody scribbled them in here
Darn it, that's gross.
previously. Error
That's a
bars,
perfect
error
spot.
bars, right?
So nasty.
Maybe not. They look very, very similar. So I think there is a little bit here. But this
I'm
is
gonna
to
buy
show you, whenever we
Hogwarts?
draw these line
Hogwarts.
graphs, there might be
Today?
some error that we're missing, and we're not
Stop,
including
stop,
it here. So we know
Can
the
you put
error is
the-
there. We
No.
know there's actually, this is like average, should be average what we're seeing here.
Dusty there.
That's drawn here as the average values, no error
No,
bars,
back up,
but
bro.
anyways. Okay, so
We're actually gonna knock her over and she won't touch it. What happened?
that's one thing I would like to highlight here, just to take this a step further. You guys all
Yeah,
get
but-
the concept about the
just
genotype and
gross,
the
like-
interaction with the environment. I'm happy with
I
that.
didn't even touch it.
One of the things is if we look at this graph, we're missing something hugely important.
That's just
Who here is
gross.
taking statistics?
Who here is autistic?
Okay, this is probably irritating
see
you
that?
to see this
Taking
graph without
statistics.
statistics on it. Okay, so, for example, you see this bar right here and how it's
Then really, I'm
slightly
gonna put
larger,
those-
it's higher, it has more
One
Yeah,
here
we-
percentage than
I
this
was
one here,
talking
right?
from
If we were to
behind.
draw a straight line across, this one's probably like
One here.
35.
And one right here.
This one's here probably like
And
30,
Deku up top.
okay? Depending on the
Or
counts,
right there.
that
up there.
could be a significant
there.
difference or not.
Yeah. Deku?
The numbers of people
This
where they
was a
get
waste of
this, it
money.
really influences,
No.
and
so
statistics
good.
is
Every
kind of
night.
like that's, in science generally,
Every night you
that
guys
tells
are
all.
night.
Okay, so if these were found to have a statistical difference, then we would consider them different. If there was no statistical difference, which there likely is
What
not,
dog?
maybe there is, but there's likely not. Why do I say there's likely
bit.
not?
Just sit
Because
right
the error
there.
bars
And two mine's gonna.
are overlapping between this one and this one. But again,
Stop.
the number of individuals
Just look
counted
right at it.
may mean that this is a significant difference.
Yeah,
to this.
but-
So I guess what I'm saying here is we don't have statistical values shown on this graph to even show that this value
don't wanna
here
be there,
is
bro.
significantly larger than this one
Falling
here.
towards
We're
it.
just assuming it is. Best
It's
would
actually so
be
gross.
if we actually had statistical values
Yeah.
showing us.
How you
That it
leave
is
marks? That's-
Okay,
Yeah, I've noticed that for a while.
I'm gonna finish it off
No,
there.
it's probably
If I don't see you on Tuesday,
Tuesday.
have a great, relaxing break.
Tuesday.
But
Today's
Next
keep in mind, there's
Really?
nothing due over the break. Have a good time, but don't forget about this stuff before the break comes, please.
Want midterm?
Okay? All right.
Midterm?
No. Something else.
There's only 5 people on the Zoom call.